Welcome to the Bodhi Academy. My name is Amisha Kulaso, and in today's video we will continue with our chapter The Fundamental Units of Life. So previously in this chapter we spoke about the nucleus. So we said that nucleus, it has a double layered covering that is called as the nuclear membrane. This nuclear membrane has this tiny pores which helps in transfer of material from inside to outside of the nucleus, that is into the cytoplasm. We also said that the nucleus, it contains chromosomes which are rod shaped structures, and only when the cell is about to divide, these chromosomes, they contain the information about inheritance of characters from parents to the next offspring that is coming. It does so in the form in the generation in the form of DNA, that is deoxyriboxynucleic acid molecules. Then chromosomes are composed of DNA and proteins. These DNA molecules, they contain necessary information, and segments of DNAs are known as the genes. Now the nucleus, we said, it plays a central role in a cellular reproduction, that is the process by which a cell divides and forms two separate new cells. It also plays a crucial role along with the environment in determining the way the cell will develop and what form it will exhibit on its maturity. In some regions like bacteria, we spoke that the nuclear region of the cell may be poorly defined due to the absence of the nuclear membrane. The one where there is absence of this nuclear membrane, such organisms are called as the prokaryotic organisms, and the ones which have this nuclear membrane are called as the eukaryotes. Now these prokaryotic cells, they also lack the cytoplasmic organelles which are there, whereas the eukaryotic cells have the cytoplasmic organelles. Many of the functions of such organelles are also to perform by poorly organized parts of the cytoplasm. Also, the chlorophyll in photosynthetic prokaryotic bacteria is associated with the membranous vessel, but not with the plastids as in eukaryotic cells. Then further we looked at the cytoplasm, and we said that the cell organelles, so when we look at the temporary mounts of onion peel as well as human cheek, we can see large region of each cell enclosed by the cell membrane. This region, that is the inside the cell membrane, is known as the cytoplasm, and this cytoplasm which is there, it often tends to have very little stain. So the cytoplasm, it is a fluid content inside the plasma membrane. It also contains some of the specialized cell organelles. Each of these organelles, they perform different specific functions for each of the cells that are present. These cell organelles, they are enclosed by the membranes. In prokaryotes, besides the absence of the nuclear membrane, they also don't have this membrane bound cell organelles. On the other hand, the eukaryotic cells, they have the nuclear membrane as well as the membrane bound organelles. The significance of membranes can be illustrated with the help of various viruses. Then we spoke about the cell organelles. Now every cell, it has a membrane around it to keep its own content separate from the external environment that is present. Large and complex cells, including cells from multicellular organisms, they need a lot of chemical activities to support their complicated structures and functions. Now to keep these activities of different kinds separate from each other, each cell is having a membrane bound little structures within themselves. This is one of the feature of the eukaryotic cells that distinguishes itself from the prokaryotic cell. Some of these organelles are visible only with the electron microscope. Now some important examples of these cell organelles are what we are going to discuss in today's video. So in today's video we will talk about the endoplasmic reticulum, the golgi apparatus, and the lysosomes. So let's begin with endoplasmic reticulum. So the endoplasmic reticulum is a large network. It is a large network of membrane bound tubes and sheaths. It looks like long tubules or round or oblong bags The ER membrane is similar in structure of the plasma membrane. So firstly, endoplasmic reticulum or ER, it is a large network of different kinds of membrane bound tubes and sheets which are present. So they are present in the form of some kind of tubes or flat sheets. They look a lot like long tubes, or even they look circular or like oblong bags, that are the tiny vessels like kind of thing which are present. So this ER membrane, it is similar to, it has a structure that is similar to that of the plasma membrane. Now endoplasmic reticulum is further divided into two categories. One is the rough endoplasmic reticulum and the next is the smooth endoplasmic reticulum. Now talking about the rough endoplasmic reticulum, firstly it looks rough because it has these particles called ribosomes that are attached to it. So it has this thread like structures outside, not exactly thread like, but it has some external features like thing that are like tiny tubes coming out. That is the reason why it looks rough, because of these particles which are known as the ribosomes. Now what are these ribosomes? So these ribosomes, they are present in all the active cells, and what they do is that they manufacture the various proteins. These manufactured proteins are then sent to various places depending on the need using the endoplasmic reticulum. So under rough endoplasmic reticulum, we know that it is rough because it has particles called ribosomes that are attached to it. Secondly, these ribosomes are present in the active cells where there is a manufacturing proteins that happens. Once they have manufactured the protein, they are sent to various places depending on the need using the endoplasmic reticulum. Then we have the smooth endoplasmic reticulum. So this smooth endoplasmic reticulum, it helps in the manufacture of fat molecules or lipids, and these are important for various cell functions. Now there is another process that takes place over here, that is known as the membrane biogenesis. Now what is this membrane biogenesis? So whatever fat molecules or lipids are being manufactured here, okay, they help in the building of the cell membrane, and this process is known as the membrane biogenesis. So that was the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. Now although the ER, that is the endoplasmic reticulum, varies greatly in appearance in different cells, it always forms a network system. So this ER is never having a fixed appearance. It can be it can have different appearances in different cells, but it will always be in a network structure. You won't anytime see it as a single cell. So it will always form a huge network kind of a system. Now what are the functions of this endoplasmic reticulum? One function of ER is to serve as channels for transport of materials, especially proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. So what we had discussed was, once the manufacturing of protein happens, then they are transported to various places depending on the need. So now this ER also helps in the transportation of these materials, especially the proteins, between various regions of cytoplasm or between cytoplasm and the nucleus. The ER also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. So it acts as a cytoplasmic framework wherein it provides a surface for many of the biochemical activities that take place in the cell. In the liver cells of the group of animals called vertebrates, smooth endoplasmic reticulum plays a crucial role in detoxifying many poisons and drugs. So in this ER, that is the endoplasmic reticulum, it is present in the liver cells of the group of animals that are known as the vertebrates. In that case, this smooth endoplasmic reticulum that is there, it helps in detoxifying many of the poisons and drugs that are present into their bodies. Now next, we will talk about the Golgi apparatus. Now it was first described by Camillo Golgi. It consists of a system of membrane bound vesicles, that is flattened sacs arranged approximately parallel to each other in stacks called cisterns. So what is happening? It consists of a system of membrane bound vesicles. In this, they are like flat structures that are there, and they are arranged approximately parallel to each other. So if one is here, the other one is going to be here, and they are stacked up into, and this they are stacked up into this high level molecules. So in that is called as this cisterns. So the material synthesized near the ER is packed and dispatched to various targets inside and outside the cell through the Golgi apparatus. Now whatever material or whatever proteins or lipids are being manufactured in the ER, they are packed and they are dispatched to various targets inside and outside the cell using this through this Golgi apparatus. Now what is the function of this Golgi apparatus? Its functions include the storage, modification and packaging of products in vessels. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus. So first is the storage, modification, packaging of products in the vessel. Second is that some of the complex sugars that are there, they are made from the simpler sugars that are present in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. Now what are these lysosomes? So lysosomes are membrane bound sacs filled with digestive enzymes. These enzymes are made by rough endoplasmic reticulum. So lysosomes are membrane bound sacs which are filled with these digestive enzymes that are present. Lysosomes are a kind of waste disposal system of the cell. Just like how a human body has a waste disposal system that is working, similarly each of the cells have these lysosomes that act as the waste disposal system of the cell. They help in to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell such as a bacteria or food as well as other organelles end up in the lysosome. So any material that is not supposed to be present into the cell, that is say that any kind of bacteria or any kind of food material that has reached there, this everything ends up in these lysosomes, which break complex substances into simpler substances. So these lysosomes, then they break down these whatever substances are there, the complex ones, they break them into a simpler substance. Lysosomes are able to do this because they contain powerful digestive enzymes which are capable of breaking down all organic material. So why only lysosomes? Because they have this powerful digestive enzymes which are able to break down all the organic material. Now lysosomes are also known as these suicide bags of cells. So we will see why they are known as suicide bags of a cell. So during the disturbance in a cellular metabolism, for example, when the cell gets damaged, the lysosomes may burst and the enzymes digest their own cells. Therefore, lysosomes are also known as suicide bags of a cell. So what is happening is that during any kind of disturbance in the cellular metabolism, say the cell is damaged or there is some kind of disturbance that is created in the cell, the lysosomes that are present, they may burst. Okay, on bursting, what happens? The enzymes that are present in this lysosomes, they digest their own cell. Therefore, the lysosomes, they are known as the suicide bags of a cell. Okay, so in today's video, we spoke about the endoplasmic reticulum, that is, we said it is a large network of membrane bound tubes and sheets. It is similar to the structure of plasma membrane. Further, we divided it into rough endoplasmic reticulum and smooth endoplasmic reticulum. We said that it looks rough because it has the particles known as ribosomes that are attached to it. So these ribosomes, they are present in the active cells and the sites of protein manufacture. Whatever active cells are there, these ribosomes are present over there and they help in the manufacturing of the proteins. Once the manufacturing is done, these proteins are sent to various places depending on the need using this ER. The smooth endoplasmic reticulum, we said it helps in the manufacture of fat molecules or lipids which are important for cell function. Then there is also another process that is taking place over there, that is membrane biogenesis. And what happens in this process is that whatever lipids or proteins that are being generated or manufactured, they help in the building of cell membrane, and that process is known as membrane biogenesis. Some of the functions of ER, firstly we said that it serves as a channel for transportation of materials like the proteins and lipids between the cytoplasm or between the cytoplasm and the nucleus. Second is that it functions as a cytoplasmic frame, providing a surface for some of the biochemical activities that take place in the cell. Thirdly, in the liver cells of groups of animals such as vertebrates, this SER, that is smooth endoplasmic reticulum, they play a major role in detoxifying many of the poisons and drugs. Then we spoke about the Golgi apparatus. We said it consists of a membrane bound vesicles that are arranged parallel to each other, and they are stacked up into this stacks called as the cisterns. The material that is synthesized near the ER, it is packed and dispatched to various targets inside and outside the cell through this Golgi apparatus. Some of the functions of Golgi apparatus: first is storage, modification and packaging of products in the vesicles. Second, in some cases, whatever the complex sugars are there, they are made from simpler sugars in the Golgi apparatus itself. Lastly, Golgi apparatus, it is also involved in the formation of lysosomes. Now, what are lysosomes? Lysosomes are the membrane bound sacs that are filled with digestive enzymes. These enzymes are made by the rough endoplasmic reticulum. These lysosomes are kind of a waste disposal system of the cell. So they help in keeping the cell clean by digesting any foreign material as well as a worn out cell organelles. Further, whenever they see a foreign material that is entering, say a bacteria or a food, what happens is that the lysosomes, they break these complex substances into simpler substances, and they do so because they have these powerful digestive enzymes that are capable of breaking down all organic material. Lastly, we said that lysosomes are also known as suicide bags of a cell. Why so? Because during the disturbance of the cellular metabolism, say the cell is disturbed or the cell is damaged, in that case the lysosomes, they burst open and the enzymes which are present inside, they digest their own cell. Therefore, the lysosomes are also known as the suicide bags of a cell. Further, in the next part of the video, we'll talk about the mitochondria, plastids, vacuoles and the cell division. Thank you so much for watching today's video. Please don't forget to like, share and subscribe to the Bodhi Academy.